Welcome to Lesson 12.3, DNA Replication. In this lesson, we'll summarize the events of DNA replication and compare DNA replication in prokaryotes with that of eukaryotes. Let's review the DNA structure. Draw this in your notes and label them with the correct parts. As you recall, the DNA structure is a double helix, or two twisted strands around each other. If you were to untwist the strands, it would look like the ladder. On the outside of the ladder would be the sugars and phosphates backbone, or this blue area. The rungs of the ladder are made up of the nitrogenous bases adenine, guanine, cytosine, and thymine. And as you recall, adenine is complementary or pairs with thymine, and guanine is complementary to cytosine. And the overall shape of the DNA structure is known as a double helix. Think back to the cell cycle. Recall the phase where the cell copied its DNA. That process is called DNA replication. DNA replication occurs during the S phase of the cell cycle. So why does a cell replicate its DNA? Well, it needs to copy its DNA so that the resulting daughter cells each have a copy of the parent's DNA. On the screen is a picture or micrograph of some DNA replicating during the S phase of the cell cycle. The process of DNA replication is aided by enzymes known as DNA polymerase. The enzymes unzip the DNA strands, and as they replicate, they proofread their work, making sure that the resulting DNA strands are identical to the parent. Let's take a look at this drawing of replication in process. Notice that one side of the DNA strand is blue, while the other side is red. The blue strand represents the original or template DNA strand, and the red strand represents the new strand. DNA replication is described as semi-conservative because half of the old DNA is conserved in a new strand. Telomeres are the tips of eukaryotic chromosomes, and they are difficult to replicate. Cells use a special enzyme called telomerase to add short, repeated DNA sequences to telomeres as chromosomes are replicated. The micrograph here shows human chromosomes in blue, and the tips are stained in white. Those are the telomeres. Unlike eukaryotic DNA, the DNA of prokaryotic organisms exists as a single loop. Replication in most prokaryotic cells begins at a single starting point and proceeds in two directions until the entire chromosome is copied.